Personalized gene editing might have just saved this baby's life. It definitely changed it. This is KJ. KJ was born with a metabolic disorder that means that ammonia builds up in his blood. It can be fatal without a liver transplant, and KJ's case was so bad that he was put on the liver transplant list at just 5 months old. But right after birth, doctors and scientists sequenced KJ's genome. They found that he had single nucleotide variants, or small changes in his DNA, in a gene called CPS1. This gene makes one of the enzymes responsible for breaking down ammonia in the blood, and KJ's variants meant that his version of the enzyme was formed incorrectly, so it couldn't do its job. So the team of doctors and scientists wanted to change one of KJ's variants, changing it from a DNA typo into a functional copy of the gene. And they tried to do this using a special type of CRISPR gene editing called a base editor. This can zip into the genome and specifically turn one base into another, here turning a T into a C. And they did this so fast. They developed a cell line to test this in vitro, they developed a mouse line to test this in vivo, they did tons of sequencing, they did toxicology tests, they submitted this for approval, and they were able to get to a first dose within 8 months. That is incredibly fast to get to a new therapy and speaks to the huge amount of work that this entire team put in. But did it work? After receiving 2 doses of the gene editing therapy, the team was able to increase KJ's protein intake. This is something that indicates that he was better able to process ammonia. They were also able to decrease some of the medications that he was on. He was also showing some clinical signs of improvement, and altogether this seems like he now had some amount of functional enzyme. That would indicate that this therapy seems to have worked. But there are a lot of remaining questions and caveats. First, one of the things that made this really feasible is that the genes that needed to be edited were the genes in liver cells. Your liver filters basically everything in your body, so it's really easy to get therapies like this to the liver, because if you put it in the blood, it's gonna get there. That's not the case for other tissues like your brain or muscles, so they had a good starting target. But because they don't want to do a liver biopsy at this point, because KJ is still a tiny baby where the risk of that just doesn't outweigh the knowledge we would gain, we don't actually know what percentage of genes were changed in the liver. The therapy could have reached 100% of the liver cells, 50% of the liver cells, 10% of the liver cells. We actually don't know yet. But there will likely be long-term follow-up for these kinds of things to see how effective and durable the treatment was. Also, although they don't put a specific price point on this is not cheap. This is 8 months of work from a whole team of people, cell lines, mouse lines, a whole bunch of testing. I don't know what that number is, but I would put this at like a 6-figure endeavor. This isn't something we currently know how to fund for every patient that needs it. Though I would like to point out that this research was funded by a number of NIH grants. This is the kind of work that federal funding supports. The kind of work that can change a child's life. And all the caveats aside, this is still a huge advance. From diagnosis to personalized gene editing therapy in 8 months is unheard of. This framework could be the foundation for learning how to do this for more and more people who need it. But to do that, we need to keep funding science.